Hi, everybody. So day two, we are going to talk about nuclear division and cell division. So we introduced the cell cycle yesterday, and now we're going to actually talk about the steps that are taken for your cells to actually divide. So if you remember, the cell spends the majority of its life in interphase, which is basically this preparation for the cell to divide, but 90% of its lifetime is spent in G1, S, and G2. So in G1, if you'll remember, is gap one, or basically growth phase one, where the cell is doing its daily activities and growing. It's the first stage of a cell's life. The S phase of interphase is when DNA is replicated. So we copy DNA in advance because when we talk about this type of cell division, we are making identical copies of cells, right? So you need to have the same DNA in your daughter cells that you did in your original parent cell. Okay, so DNA is actually copied before mitosis, before this nuclear and cellular division. Excuse me. And then finally you have G2 phase, which is your final preparation before we enter mitosis. So now we're going to talk about the M phase, which the M phase does encompass mitosis, which is nuclear division or division of the nucleus, and cytokinesis, which is cellular division or basically the division of the cytoplasm. So mitosis is basically your nucleus going from one nucleus to two separate nuclei, with, again, your DNA separated out, identical DNA in each of those new nuclei. And then cytokinesis is actually division of the cytoplasm or the separation of one cell into two. And again, mitosis is what your cell spends about 10% of its time doing. The rest of the time it's not really preparing for this. And just as a reminder, in your books on page 133, there is a typo in that bottom paragraph where it starts talking about the mitotic phase. Just remember that interphase is 90% of a cell's life typically, and the M phase is 10% of the cell's life. So when we talk about mitosis, we're going to break it down into four main phases. We do have a fifth intermediary step here that's technically a part of prophase, but we will break it out a little bit. You can remember these four phases in order as PMAT. So we have prophase, metaphase, anaphase, and telophase. So these four phases, or four main phases, are the steps that it takes to divide one nucleus into two, right, or nuclear division. And then at the end of telophase, we'll have cytokinesis, which is the actual separation of one cell into two. By the end of this, the next couple days, you're going to know all these phases, what happens in each step, and you'll also be able to recognize them by pictures. So this picture might not make a lot of sense to you right now, but these are actually different cells in various stages of the cell cycle. So by the time you familiarize yourself with all of these different stages, you should be able to identify cells by what their pictures look like, what stage they're in. Okay, so let's start with prophase. So prophase is the first step of mitosis, the first step of nuclear division. So after a cell leaves G2, it's going to enter into prophase. Now, if you'll remember, your DNA is in the form of chromatin during interphase, right? So it's loosely jumbled, looks like spaghetti. But if you try to take a bowl of spaghetti and saw that spaghetti in half, there's no way that you can guarantee that you're going to get equal amounts or identical copies of that spaghetti in two subsequent bowls, right? So in order for us to ensure that the copied DNA that, again, we copied in interphase will actually migrate to separate sides of the cell and end up in two different subsequent daughter cells, we need to condense the chromatin into chromosomes, which is a super organized form. And again, this will allow us to actually separate the DNA and ensure that we get your sister chromatids, your identical copies, one in each of your subsequent cells. Because remember, this chromosome has two twin sister chromatids, which are identical to each other. One sister chromatid is going to end up in one daughter cell. The other sister chromatid is going to end up in the other daughter cell, ensuring that this DNA is in each of the subsequent cells, right? And that's true for all four of these chromosomes that we're looking at here. Okay, so a couple main things for early prophase. Your centrioles, which are your organelles that we talked about earlier, just as being involved in cell division, but we didn't get much more into detail than that. Centrioles are made of microtubules, right? And they're present in animal cells, but not in plant cells. This is an animal cell we'd be looking at. So what actually happens here is your centrioles typically hang out near the nucleus. But at the beginning of prophase, they're actually going to start to migrate to the poles or separate ends of a cell. So you see here how they're migrating to poles. Again, this is because the centrioles are actually what's going to be involved in separating out your chromosomes and pulling them to separate ends of the cell to ensure that we end up with two daughter cells that are identical to the original and have the information that we need in each. So you can see here that your centrioles, they come in pairs. Typically, they hang out in pairs. So you have one pair of centrioles here, another pair of centrioles here. From this region, which we call the centrosome, we also get spindle fibers, which are also microtubules that are going to begin to sprout from this area. So these spindle fibers are going to play an important role because they actually are going to attach to the sister chromatids and separate them and pull them to opposite poles. So these spindle fibers in this prophase step actually start to sprout from the centrioles in the centrosome region. Okay, so you can see them sprouting here. So when we refer to the spindle, you might hear them referred to as spindle fibers or microtubules, spindles, spindle microtubules, all the same thing. Okay, and again, when you have centrioles plus spindle fibers, this is your centrosome. So centrosome technically refers to a region where you would find both of these here. Another thing that occurs in prophase is that your nucleolus is going to disappear. So again, we actually need to divide this nucleus. So we actually need to temporarily break it down. So the nucleolus will temporarily disappear. We do not need it for these things. So that will just dissipate, and then it'll show up when we get to the end. You'll also notice this here that looks like it's starting to break apart. This is your nuclear membrane. So we're going to refer to this more in the next step that we get to in late prophase. But you can see that the nuclear membrane is actually starting to break down a little bit because, again, we need to break down the nuclear envelope in order to separate out our chromosomes, right? We're actually not doing that within the boundaries of a nucleus. We need to temporarily break down that nucleus. So here's just a picture of early prophase in a cell. Okay, so let's talk about prometaphase. So this is technically late prophase, but it's a transition step between prophase and metaphase. So in prometaphase, right, you have a few major things occurring. One, your nuclear envelope that I just was talking about in early prophase is going to completely break down and disappear by the end of prometaphase. It will be dissipated, so you won't be able to see the nuclear envelope any longer, which again is the same thing as the nuclear membrane. You also get your microtubules that we just referred to, those spindle fibers. They're going to begin again, so these centrosomes and centrioles will be migrating to opposite ends of the cell. And then these spindle fibers are actually extending towards your chromosomes because these spindles are going to attach to each of your two sister chromatids. Now, you'll notice on your sister chromatids, so here's one twin and here's the other twin, right? This is copied DNA. This is one identical copy. This is another. Each of these sister chromatids has what looks like a landing pad on them. That's called a kinetochore. So kinetochore is basically a protein complex. I think of it as a landing pad for these spindle fibers or microtubules to attach. So these spindle fibers are going to attach to the kinetochores, which are in the centromere. A lot of CENT words, so make sure you get those cleared and that you practice your vocab. But your kinetochores are found in the centromere region, which again is that center region of a chromosome. And again, the kinetochores are basically landing pads where these spindle fibers are going to attach to, because the spindle fibers are going to be involved in actually pulling your sister chromatids apart from one another. Okay, so by the end of prometaphase, your spindle fibers are actually, your centrosomes are going to be, again, in opposite poles. Your spindle fibers will begin attaching to your kinetochores, or again, they might attach to each other. So you have a lot of attachments occurring here, and basically these chromosomes are going to start migrating towards the middle of the cell. So in metaphase, I want you to think middle. So metaphase is super easy to recognize in pictures because at this stage, you have all your chromosomes lined up in the middle of the cell, or at basically the equator of the cell, right? So here your spindle fibers are fully formed. You can see that some of those spindle fibers are attached to your kinetochores. Again, you're going to have two attachments, right? One for each sister chromatid. And then the kinetochores will face the poles that they're going to be pulled towards, right? So again, metaphase, you have spindle fibers are fully attached. Your chromosomes are lined up in the center of the cell. So here's a good picture of metaphase, again in what looks like a plant cell, right? Here's another cool picture of metaphase here. So you can see these are the chromosomes that are lined up, and these spindle fibers look, they look like kind of like nerve endings, but these are spindle fibers that are attached to your chromosomes. Okay, anaphase. So the third step of mitosis. Think away, okay, or apart. So when we're talking about anaphase, we're talking about chromosomes moving away from one another. The sister chromatids are being pulled apart and moving away or apart towards the poles. So here is where your spindle fibers, right, are actually going to start pulling your sister chromatids from the kinetochore region where they're attached. So you can see your spindle fibers are attached here. Now these two twin sisters, which were again bound to each other in the centromere region, have now been pulled apart and are being pulled towards opposite poles. So basically the microtubules are going to begin to shorten to kind of reel those to opposite ends, right? The cell is also going to begin to elongate at this point because again, our end goal is to make 2 daughter cells where originally we had 1. So here are a couple pictures of anaphase. So again, you can see the chromosomes are moving away from one another. The sister chromatids have been pulled apart. Now telophase. So here your cell's going to elongate even more. So telophase is basically the opposite of prophase. So everything that we did in prophase to temporarily break down the nucleus, condense chromatin into chromosomes, all that's going to be reversed in telophase because basically now we've separated out our sister chromatids. So we have identical DNA in this side of the cell and this side of the cell, right? So now our nuclei are going to reform. So you're going to get a nuclear envelope that reforms around the DNA here, a nuclear envelope that reforms around the DNA here. Because again, our goal was to make a copy of our original nucleus, right? So that's what we've done here. So basically you have the nucleolus that will begin to show up as well. And again, the cell is going to continue to elongate in preparation for that cellular division. Your chromosomes will also decondense. They'll begin to decondense into chromatin again at this point as well. And here are a couple cool pictures of telophase. Now telophase, you can see here, so here are now the 2 separate nuclei and the cell is beginning to split into 2. So cytokinesis is our final step where we actually get the division of the cytoplasm or the division of the cell, right? This typically happens simultaneously with telophase. So we say, you know, it might happen at the end of telophase, but it's really typically happening simultaneously. Now what's happening here is you can see in telophase, again, your nuclear envelopes are reforming, right? Your spindle fibers have been reduced, centrioles, are going back to just hanging out. And again, your DNA is decondensing from chromosomes to chromatin. And cytokinesis is the actual division of the cytoplasm or the making of 2 cells from 1. So we'll talk about the difference between animal and plant cells here, but this is an example of an animal cell. You can see we actually have a pinching inward or a cleavage furrow that's forming here. So it's basically pinching in to separate out. And again, your 2 daughter cells that you end up with are identical to your original parent cell that you started out with, and they're identical, of course, to each other. Sorry, one more note on that. I just want to remind you, so when we talk about mitosis, we're talking about asexual reproduction because this is the process that all of your somatic cells or body cells undergo for growth, for repair, and for replacement, right? So this is all making clones or identical copies of your cells in order for you to grow, repair, replace. Okay, so just a brief overview of cytokinesis. I'm going to try to wrap this up for one video in 15 minutes. So in animal cells, cytokinesis happens a little differently than in plant cells because we only have a cell membrane, whereas plant cells have a cell wall. So in animal cells, we only have our cell membrane. You're going to get this pinching in, or what we call a cleavage furrow that occurs. Basically, the cytoplasm and cell membrane is going to pinch inward until you actually separate the 2 cells. So we call that a cleavage furrow, again, when you have this narrowing in or pinching in of the cell, right? And then it'll eventually pinch apart. Now, plant cells can't do that because plant cells have cell walls, and they're not actually going to pinch apart from one another and then just go float off totally separately. You know, as we know, plant cells are super rigid and they're very close together. So what happens in plant cells is we actually get the formation of a cell plate. So cellulose, right, which is what our cell walls are made of, is actually going to be pulled to the center in between plant cells and build up in between those cells. So here's an animal cell. And here's a plant cell.